Okay, having looked at the various reactions of PLP, now let's look at how we break down some particular amino acids. What I want us to recognize first of all is that a nice endpoint is pyruvate. Pyruvate, that's sort of, I mean, if we think about where we want to break these down to, let's get to some things that are useful for other processes. So we have pyruvate, we have oxaloacetate, we have alpha ketoglutarate, we have succinyl-CoA, so alpha ketoglutarate, I guess I can draw that. That's not a big deal. We've looked at that a few times. All right, all the molecules. Succinyl-CoA, and we can get to fumarate, and then we can get to acetyl-CoA, all right, goodness, I didn't think this would be that big. And then we have acetyl acetate. Okay, so all of these structures are nice endpoints as we're looking at what we're going to do with the carbon atoms within our deaminated, decarboxylated, retroaldol, retroclasm, all those reactions that PLP can manage. If we can get to any of these, we're in good shape. Why are we in good shape? Because all of these are involved within the citric acid cycle. And so they're productive for other important metabolic processes in the body, as opposed to like one-off metabolites that we sort of have to deal with. So we have our carbon atoms ultimately ending up in something that can be used within the citric acid cycle. Okay, then we can classify these things as, you know, if they form keto acids or the equivalent of keto acids, we can call them ketogenic amino acids, glucogenic amino acids, ketogenic amino acids or glucogenic amino acids. There's a nice discussion of that. There's probably some relevance to like the keto diet and that sort of thing. Let's not go there. Okay, so what I want to do is just let's start with all the PLP reactions in mind and with some idea of what's an important stopping point within within the catabolism of the amino acid side chain. Let's start to look at these. Let's start with alanine. Alanine is a great one to start with because given what we've seen, it's actually fairly simple. What you can do is if you take this and react it with PLP, alpha ketoglutarate, we can do a transamination reaction. And a transamination reaction is just going to convert the NH2 into a keto. And anytime we can just do that and make pyruvic acid, which has a negative charge is pyruvate, we're done. Consider it catabolized or catabolized. Now, if we take serine, we can do something similar. But in this case, what I want to do is I want to adduct that with PLP and then do a dehydration reaction. Now, dehydration means we're going to deprotonate the alpha position and kick off the OH to get minus water. And when we do this, all we have to do is recognize that we have an enamine. So enamines are like imines. We've talked about them a little bit in organic too, but enamines are like imines in that when they see a carbonyl, they're going to go back to the ketone plus the amine that they started from. Okay, so when we react this, we're going to replace the nitrogen with an OH. And this is sort of like a transamination reaction, but it's after a dehydration. So we didn't start with the same amine that we went in with. And this is just the enol form of pyruvic acid again. And because I've left this deprotonated, I guess I'm going to have to call this pyruvate. Pyruvate, pyruvic acid, the same. We're into the citric acid cycle. We're done. Just by using those common reactions of PLP. Okay, what do we want to do next? Well, we can consider an alternative strategy of dealing with serine, and that gets us into glycine metabolism, which is an important consideration. So an alternative to serine dehydration. That is, let's go ahead and try a retroaldol reaction. So with the retroaldol reaction, what we do is we take serine and we deprotonate the OH to push the negative charge into the alpha position. I guess that's no longer chiral, but at this point now, we can pick up H plus and we call this the glycine adduct with PLP. Why do I call it that? Well, if you look at this framework, this looks like an amino acid adducted to PLP, but we have two hydrogens at the alpha position. That's just like what we would get with glycine, except we've gotten here from a retroaldol reaction of serine. So we could add some water if we wanted to get to glycine. But I want to hold on to this idea. Yeah, we can make glycine from serine. That's fine. Okay. We can make, and we typically would prefer to make serine from glycine as opposed to making glycine from serine. But anyway, what I want to do is just intercept, stop here. Stop this thought process, not stop the lecture. In thinking about glycine PLP, we're going to revisit that in a second, but I want to just revisit what happens to formaldehyde. Nobody likes to have formaldehyde present in their body. It does not feel good. It's like a hangover molecule acetaldehyde times cranked up to 11. It makes you feel miserable. But what you can do is you can use tetrahydrofolate, which we talked about from a previous lecture, adsorbs formaldehyde. Sorry, spacing. Formaldehyde to make the methylene cofactor. Okay, so, and then that's useful for other metabolic processes that we have looked at. But what about glycine? So how is glycine metabolized? I'm going to focus on catabolize so that we can focus on the breakdown of glycine. Okay. What I want to do is go back to the glycine PLP adduct. What we're going to look at is, of the various PLP reactions, we're going to go to the decarboxylation reaction. So we're going to lose CO2 to generate this negative charge. Okay. Which is going to kind of pick up a hydrogen at this position, pushing the double bond into, or excuse me, kind of on the outset of the molecule. And this is actually an equivalent of PMP adducted to formaldehyde. So we could lose this species. We could lose this species in such a way that we regenerate some formaldehyde. But what ends up happening is we're going to react with lipoamide, the or just, I think it's just lipoamide is what it's called. And we're going to go from this negative charge here. So we're going to interact this with lipoamide. Lipoamide was a weird kind of cofactor that had two sulfur atoms that were bonded together through a disulfide bond. So what I'm going to do is I'm going to have the negative charge interact with this sulfur atom, pushing the electrons from the disulfide over to reach out and grab an H+. Okay? Now, when this reaction occurs, we're going to form an intermediate where we still have an imine to PLP, which means we don't have to worry about turning over the PMP. We have a bond to sulfur, which is 5 atoms away from an SH, which is adducted to this R group. Now we can add water to this, and that's going to cut this to regenerate the PLP aldehyde and an NH2 off of that sulfur thing. So let's go ahead and just drop the entire pyridoxal phosphate cofactor. And let's see how we're going to deal with this. Species. Well, the book says that this will lose NH3 and we actually add the tetrahydrofolate to this. That's THF. We'll lose NH3. We'll lose the methylene, THF, and we'll get this dihydrolipamide, which we've talked about previously as another metabolite that we can deal with. So what we end up though getting is we end up converting this into an ammonia and formaldehyde equivalent. So we've gone all the way down to formaldehyde, but it's baked into that cofactor that can stabilize and actually use formaldehyde in low dosages, along with NH3, which can get dealt with in some urea cycle processes. Okay, chugging along through, we can go to cysteine now. So cysteine metabolism. So cysteine actually doesn't need to bind to the PLP cofactor, it appears. Instead, what cysteine is going to do is rely on its innate proneness. It's innately prone to getting oxidized at sulfur. I've actually made, if you make a non-zwitterionic cysteine, that is, you protect the nitrogen so you can't exist as zwitterion anymore, then you get an oil. The oil can more efficiently interact with gases like oxygen from the atmosphere, and you'll see that sulfur oxidize very rapidly. In the body, you can do probably an enzyme catalyzed oxidation. Sulfur is just so prone to oxidizing, and here we're going to take it to the sulfinic acid. Don't worry about the I guess I'll just put the nitrogen up. Don't worry about the sulfinic acid or the sulfonate name. That's me doing my PhD with some sulfur chemistry. But anyway, this is now able to react with PLP and alpha keto glutarate, and we do a transamination. The transamination reactions convert alpha amino groups into alpha keto groups. And the sulfonate actually sees a nice opportunity to lose SO2 to generate the enolate of pyruvate. So we're going to lose SO2 and generate this enolate structure, which can grab an H+ to form pyruvic acid. And we're into the citric acid cycle. Okay, so with threonine, we're going to do yet another different type of PLP reaction. And for the case of threonine, we're going to rely on the Retroaldol reaction. We can actually do some stuff like what we've seen before, where we can do Retroaldol reactions, and we can even do we can even intercept other pathways. We'll talk about that. Let's start with the Retroaldol reaction is the newest one. So in the Retroaldol reaction, we take threonine, which is a beta hydroxy amino acid like serine, except that gamma position also is present with the methyl group. What we're going to do is adduct that with PLP to form our PLP imine, an external aldimine within a PLP-dependent enzyme. And then we're going to react this with NAD+ to do an oxidation. Remember, when we don't write the H, that means it needs an H, which means that gets reduced, which means our substrate gets oxidized. So we're going to oxidize the beta keto position, or excuse me, the beta hydroxy position to a beta keto group to make this species. And now what we're going to do is engage this with CoAsh, or the CoA-free thiol, and that will get deprotonated. But what we can do is add to the beta keto, push up, push down, and then do the Retroaldol reaction to generate the glycine anion. That's a little sloppy. I'm going to clean that up. We can put the negative charge at the alpha position of our amino acid that's adducted to PLP. But this is the glycine anion after it's adducted to PLP. And we get this as a result after the sulfur has added, and this is just acetyl-CoA. Whereas what we can do with this PLP adduct of glycine, we can add H+, we can add H2O, and that will give back glycine. And maybe glycine's what we wanted. Maybe glycine goes through the glycine metabolic pathways we just touched on, but we regenerate PLP, and we have dealt with that side chain because acetyl-CoA, if you recall, is a super important molecule in all of biochemistry. And it's directly involved within the citric acid cycle. Okay, we could also do a dehydration of threonine. So threonine's a pretty versatile amino acid. Maybe threonine's my favorite amino acid. There's just so much great reactivity within amino acids. I mean, honestly, like, it's just so great. Sorry, okay, back to business. So we're gonna take threonine and already have it adducted to PLP. With a dehydration, what I need to do is let some base come, remove the alpha hydrogen, and then push off the OH to get a net loss of H2O. Okay. Now, if we do this, we have an enamine again. The enamine can react with water where we swap the nitrogen for an oxygen. Let's not worry about what's happening with the PLP. The answer is nothing important that can't be easily dealt with using stuff we've already seen. But the outcome is here we get an enol. And so we get an enol that tautomerizes to alpha ketobutyrate. It's a butyrate because butyric acid is kind of the common name of butanoic acid. So that's where that name comes from. So we get from alpha ketobutyrate. Now, alpha ketobutyrate through some other pathways can get converted into propionyl-CoA through a decarboxylation and a thioester formation. We've seen reactions like this. So this is propionyl-CoA CoA, and then propionyl CoA can go into, can do a subsequent reaction through a few series of steps to make. The book goes through this. I'm going to bypass it just because I want to focus and not overwhelm. But we're going to actually carboxylate the gamma position, and that makes succinyl CoA. It's a really cool reaction. I don't mean to, I feel like I've been long-winded this lecture. But succinyl CoA now enters the citric acid cycle, and we've hit that. So a lot of curly arrows. A lot of this involves the use of PLP, some super cool reactions. And I think this is a good breaking point. We haven't touched on all of the amino acids, right? We haven't broken them all down, but we've broken down a huge chunk of them. And so at this point, I think this is a good stopping place for the lecture. So we'll see you next time.